Hello everyone, my name is Justin Woodrow, and that's the face that goes with the voice. In this presentation, I will be discussing the ketogenic diet, how it might affect and protect the nervous system, and how it might fit in with human performance. But first, I'd like to take this time to give a special thanks to Logan University, to Dr. Conrad Woolsey, and to Commander Thomas Sather of the United States Navy. Without them, it wouldn't be possible for this internship. So I'd like to just say thank you. Now, there are some objectives that go along with this presentation. The first of which is to demystify the ketogenic diet, or the KD, which will be referred to throughout this presentation. We will be exploring neuroprotection and the preventative effects of a KD. We will be relating seizures and oxygen toxicity, and hopefully we will uncover the mechanisms affecting seizure prevention. And we will be exploring a KD in human performance. And finally, we will complement a KD with nutritional strategies. Now, this slide here shows a couple nice meals that most people would not feel like that they should be eating because it's seemingly high in fat, which is very true. So that brings us to think, well, what is the ketogenic diet? What is the KD? And it's a high-fat diet that causes physiologic changes that mimic starvation or periods of limited food availability. Now, we will be getting into the mechanisms behind how this diet is similar to those conditions, but we'll be getting to that a little bit later. So instead of glucose, ketone bodies become the body's primary energy source, and this is termed nutritional ketosis. Ketone bodies will be discussed in greater detail also later in the presentation, but for now just realize that these are the source of energy, hence the name ketogenic, the ketone producing diet. Well, the KD is composed of 80 to 90% of calories coming from fat and enough protein for growth, which is roughly 50 to 80 grams per day, but that's based on the overall mass of the individual. But this diet lacks a sufficient carbohydrate load, so you're only going to be getting about 10 to 20 grams per day. Now, basically, 0.8 grams per kilogram of protein per day is considered adequate intake of protein, and so in a 100 kilogram person, 80 grams would suffice. Now, the amount of carbohydrate intake is minimal, and as we will see, there is an adaptation of sorts to the ketogenic diet. Now, relating more specifically, there's a ratio that's commonly used. It's 4:1, and this represents the total mass of fat to the combined mass of protein and carbohydrates. This ratio can be modified over the course of the KD administration for individualization purposes, meaning based on the person and what kind of effects are trying to be experienced here. Now, I'm just going to give you an example. So if we have a 2,000 calorie diet and the KD is used, 80% of calories come from fat and sufficient protein is granted for growth, then this would basically mean there would be 1,600 calories that would come from fat, and 320 calories from protein and 80 calories from carbohydrates. So this kind of equates to 180 grams of fat, 80 grams of protein, and 20 grams of carbohydrate. If you would like to find out more about specifics to the ketogenic diet, how it can be kind of modified for your own liking, you can use this URL link there in the bottom, right there. So here we have an example ketogenic diet and its macronutrient breakdown. Basically, if you look through breakfast, lunch, snack, and dinner, you can see that it's extremely high in fat all throughout. And at the end of the day, only 2.7% of calories are coming from carbohydrates, with 83.2% coming from fat and 15.7% coming from protein. This correlates very well with our last slide and example, and you could find more about diets on paleoplan.com. This slide here is the ketogenic diet food pyramid. From this pyramid, the majority of the foods shown are relatively high in protein, which demonstrates the various options for caloric intake while on a ketogenic diet. The major dietary components are meats, eggs, cheese, fish, heavy whipping cream, butter, oils, nuts, and seeds. As you can see, the carbohydrates that are recommended are derived from fibrous fruits and fatty and non-starchy vegetables. A great example of fibrous fruit is raspberries, having nearly 12 grams of fiber per serving. The reason why fibrous fruits and vegetables are recommended is based on the diminished absorption of carbohydrates due to the fiber content of these food types, and they're also thought to increase fatty acid metabolism. This is an important part of the diet because the dieter may desire a sweet taste, and these fruits are great options for satisfying that craving. If you're asking yourself, could this diet provide an adequate supply of all nutrients? Well, the answer to that is no. So yes, there is a risk for micronutrient deficiency while on a ketogenic diet, and more will be discussed in the nutritional strategies section of this presentation. So you're probably wondering, are there variants of the KD? And yes, there are. There are 3 primary variants. The first variant is the classic KD, which uses long-chain triglycerides, or LCTs. But first, there's a 24 to 72 hour fast until ketosis is reached. And once reached, then they start to use the LCTs at a 3-to-1 or 4-to-1 ratio, like we described earlier, along with 1 gram per kilogram per day of protein and 10 to 20 grams per day of carbohydrates. The second variant is the modified KD, which uses a medium-chain triglycerides, or MCTs. The MCTs have been found to cause gastric upset. However, the MCTs allow for ketosis to be readily achieved due to their direct absorption into the blood supply, instead of the typical chylomicron lymphatic journey of dietary fats like LCTs. This allows for the consumed MCTs to reach the liver for ketogenesis in rapid fashion. Also, that allows for more non-ketogenic foods to be consumed, and it's more palatable. The modified KD is also used when serum triglycerides are elevated, typically when on a classic KD. The coconut oil is a very good option if the MCT oils cause gastric upset. However, when using coconut oil, 2 to 3 times the amount of MCT oil must be used because they're less concentrated. The Radcliffe Infirmary diet is the 3rd primary variant of the KD, which uses a mixture of LCTs and MCTs. Another variant could be considered, and that's the Atkins diet. The only true difference is that calories are not restricted like that of the classic KD, but the carbohydrate intake is still limited to 10 to 20 grams per day. I must quickly mention the cyclic and targeted KD, and these are modifications to the ketogenic diet where carbohydrates are added to afford the body anaerobic capacity during exercise. But much more will be discussed later in the human performance and exercise portion of this presentation. And finally, all 3 primary diets are thought to have similar, if not the same, efficacies. And if we mention efficacy, then there must be a reason for its use. So let's find out now. So why is the KD used? Well, for one, weight loss. The Atkins and paleo diets are commonly used to help with weight loss. And if we mention some recent news, LeBron James lost about 20 pounds, and he attributes this to his lack of carbohydrates in his diet. Another reason why the KD might be used is because of the proposed health benefits, like anti-inflammatory, anti-diabetic, and anti-obesity effects that come along with this diet. And so, if this is true, it might be helpful in the management of diabetes and the treatment and or prevention of cancer. Also, the treatment of epilepsy, historically, the KD has been used for its treatment, and a handful of other neurologic diseases as well, like ADHD, Lou Gehrig disease, Alzheimer's disease, autism, migraine, and Parkinson's disease. We will see the proposed mechanisms that relate to the treatment or prevention of some of the listed neurologic diseases, but first, let's explore ketone bodies. So what are ketone bodies? Well, they're biochemical substances that are formed in the liver from two acetyl-CoA molecules typically derived from fatty acid metabolism. They are energy sources that vary in concentration during normal conditions, calorie restriction, limited food availability, starvation, uncontrolled diabetes, and use of varying KDs, and also during prolonged intense exercise. Ketones become the primary energy source along with fatty acids during a KD. So are there different kinds of ketone bodies? Yes, and three kinds exist. The first being acetoacetate, which is technically the only true ketone body, and it can be used in most peripheral tissues. And after roughly about 5 hours, acetoacetate spontaneously decarboxylates to form acetone. So essentially it breaks down, and this use it or lose it concept attributes to the weight loss seen with the ketogenic diet, since acetone cannot be converted to acetyl-CoA or back into acetoacetate. Now, acetone, we just mentioned, and it's another ketone body as well. And this is, like we just mentioned, from the breakdown of acetoacetate. And since it can't be utilized by peripheral tissues, it however, can be utilized in the liver for energy because it's converted back into acetate. And this can only occur in the liver. But it is excreted in the urine and exhaled air. Lastly, the third type is beta-hydroxybutyrate, and this is derived from acetoacetate, and it can be used in peripheral tissues, including muscle tissue. Beta-hydroxybutyrate is the major ketone in the blood, and so according to rat studies, researchers found that beta-hydroxybutyrate made up nearly 78% of all circulating ketone bodies. And to clinically determine the degree of ketosis, beta-hydroxybutyrate levels are the ones that are measured. So why do ketone bodies form in the first place? That's a good question. First, let's briefly grasp the effects of two hormones that play a huge role in metabolism. This illustration shows the physiologic effects of insulin and glucagon within the liver. This is simply to give us a look at the general function of these two hormones. Well, in general, insulin is an anabolic hormone stimulating biochemical processes leading to synthesis and storage of molecules, whereas glucagon is a catabolic hormone stimulating biochemical processes leading to breakdown and availability of molecules. So, in a general sense then, glucose is going to be converted to glycogen under insulin's effects, and glucose will be brought about from glycogen based on glucagon's effects. And so glucagon here is going to be the focus molecule for formation of ketone bodies, and also using fats as fatty acids for energy. Although a ketogenic diet induces similar conditions seen here, there is provision of energy mainly in the form of fat, which acts to prevent protein degradation and functional tissue loss. This highlights the importance to supply some carbohydrates, as glucose is an obligate nutrient, and enough protein for cellular maintenance and growth in peripheral tissues while on a ketogenic diet. Note that there is some keto acid formation possible in peripheral cells. Earlier I mentioned that the liver produces the ketone bodies, which is true, but some peripheral tissues may be able to produce ketone bodies, as we will see shortly. Also, note the potassium loss with glucagon stimulation. This is an important ion along with sodium to consider when administering the ketogenic diet, but more to come on that later. So let's take a closer look at the catabolic state. But first, no matter how we look at it, as I repeat, glucose is an obligate nutrient which is necessary for the brain and nervous system and red and white blood cells. So, glycogen will be broken down until stores are exhausted, which takes about 18 to 24 hours for relative glycogen depletion to occur. I say relative because glycogen is never completely depleted, because some is left for a primer for glycogen resynthesis. This further highlights the fact that glucose is an obligate nutrient. These catabolic hormones will jumpstart glycogenolysis and gluconeogenesis, resulting in an increase in blood glucose. But not all glucose will be derived from glycogen, because these hormones will also utilize glucogenic amino acids from muscle protein. Alanine is the most commonly used amino acid for gluconeogenesis, which eventually becomes pyruvate and then ultimately glucose. Here we see how glucagon can affect the oxidation of fatty acids and the production of ketone bodies. So as glucagon levels rise, decreased levels of malonyl-CoA result. And malonyl-CoA is essentially a precursor for fatty acid biosynthesis, which leads to the activation of carnitine palmitoyltransferase CPT, and then eventually fatty acids enter mitochondria for oxidation and ketogenesis. It is important to note that CPT is the rate-limiting enzyme of ketogenesis. Specifically, there are two forms of CPT, CPT1 and CPT2. CPT1 groups with the activated fatty acid acyl-CoA to become acylcarnitine. Once within the mitochondrion, CPT2 reconverts acylcarnitine to fatty acid acyl-CoA. And so this process has led to the introduction of supplemental L-carnitine, with hopes that it might increase fatty acid metabolism. To date, there is no evidence to back these claims. Now, nevertheless, carnitine is endogenously synthesized using lysine and methionine, and its synthesis depends on the presence of vitamins C, B3, and B6, and also iron. So these components are definitely necessary for fatty acid metabolism. And so there's an adaptation to fasting which occurs, and so with decreased hepatic gluconeogenesis comes increased ketogenesis and more renal derived glucose. So basically the liver slows down its production of new glucose and the kidney makes up for that. Since muscles don't have an enzyme necessary to release glucose in the blood from its glycogen stores, it will, however, release pyruvate, lactate, glutamine, and alanine. The Cori cycle will eat up the pyruvate and lactate and form it into glucose. And the Cori cycle occurs in the liver, by the way. Glutamine will be deaminated, and it will be converted into alpha-ketoglutarate, which enters the Krebs cycle. And alanine, as mentioned, will be transaminated, becoming pyruvate and eventually glucose. One point to mention, though, is that once glutamine is deaminated into alpha-ketoglutarate, it has to be brought into the mitochondrion to enter the Krebs. And so malonyl-CoA is something that is utilized for bringing in alpha-ketoglutarate. So if concentrations are depleted when glucagon is elevated, then that means glutamine may not be deaminated as much. And so this may prove effective for buffering acidic metabolites in the kidney, which glutamine is known to do. So what else happens during the catabolic state? Well, lipolysis occurs, meaning breakdown of fat, due to the action of these catabolic hormones as well. And so fatty acid will be released, and its use will depend on certain hormones. And we mentioned epinephrine and glucagon already, but also cortisol and growth hormone have an effect on the release and use of fatty acids. The continuation of fatty acid metabolism via beta oxidation produces the accumulation of acetyl-CoA, which pushes the liver to produce more ketones. And we saw that with increased glucagon levels, there's going to be an adaptation to the fasting. So the liver is going to produce less glucose, and so maybe that level of glucose production affords for more production of ketone bodies. So essentially ketone body production will decrease the need for glucogenic amino acids, thereby sparing muscle protein. So we see the ketone bodies can help spare muscles from being broken down. And also, as the brain increases its usage of ketones, the need for glucogenic amino acids also decreases, which is definitely a good thing. Now, if fat stores are depleted, then protein from muscle will fuel the body. And this option is not a good one because it can lead to death. But with the ketogenic diet, there is provision of fats, proteins, and minimal carbohydrates. So this option should never become available in healthy individuals. This right here is a simplified illustration of ketogenesis. Note that the conversion of acetoacetate to beta-hydroxybutyrate is reversible, but once acetone is formed, there is no turning back. And that's what we mentioned earlier about the acetone use it or lose it phenomenon, where that's where a lot of the weight may be lost from because the acetone cannot be used again for energy, except minimally in the liver by converting the acetone into acetate. The carbon dioxide molecule yielded here during the conversion to acetone may be important, which we'll see a bit later. So this slide here is the opposite of the previous slide we saw like this, and insulin's concentration is much higher than glucagon. So basically the opposite occurs. So if insulin is administered or carbohydrates are consumed, inhibition of ketogenesis, lipolysis, glycogenolysis, and gluconeogenesis occurs. Remember, insulin is more of an anabolic phenomenon and glucagon is more of a catabolic phenomenon. Although this would ultimately negate the effects of the ketogenic diet, we will see how insulin importantly factors in with the administration of this diet. Now, one fact must be mentioned briefly, and that's the role of inflammatory mediators. Interleukin-1, tumor necrosis factor, and other inflammatory mediators stimulate insulin release. And thus, if infection or illness is present in the dieter, there may be a transient inhibition of ketone body utilization. This inflammatory scenario could also arise if too many saturated and trans fatty acids are consumed, as they are implicated in inflammatory disorders. Thus, the ketogenic diet may be useful in the prevention and treatment of chronic inflammatory disease such as type 2 diabetes mellitus and Alzheimer's disease, or more recently known as type 3 diabetes. Later in the presentation, we will look at the specific sources of fat that are best suited to keep inflammation to a minimum. This algorithm is an overview of what we just covered. So, with a ketogenic diet, starvation, calorie restriction, and uncontrolled diabetes mellitus, there will be increasing levels of glucagon and decreasing or low levels of insulin. This leads to glycogenolysis and gluconeogenesis, two mechanisms stimulated by glucagon. Glucose will be derived from glycogen stores and gluconeogenesis, with substrates coming from glucogenic amino acids and glycerol from triacylglycerols. Eventually, exhaustion of glycogen stores and glucogenic substrates occurs, creating a continuance of falling blood glucose. As glucose continues to fall and glycogen stores and glucogenic substrates become exhausted, glucagon continues to be secreted and insulin is further suppressed, leading to lipolysis and subsequent free fatty acid and glycerol release. Glycerol can be used as a glucogenic substrate due to lipolysis. But note that along with glucagon, epinephrine, growth hormone, cortisol, and thyroid hormone can stimulate lipolysis as well. As acetyl-CoA increases in concentration, pyruvate carboxylase is stimulated, which causes oxaloacetate to be formed from pyruvate. Glucogenic amino acid pools can supply the carbon backbone for pyruvate, which will occur until peripheral utilization of ketone bodies is established. Once acetyl-CoA concentrations rise to a point where they overwhelm the processing capacity of the Krebs cycle, acetyl-CoA molecules will be utilized for ketogenesis. As ketone bodies are produced, their concentration obviously continues to elevate. Eventually, the concentration of blood ketones will stimulate the release of insulin. As insulin levels increase due to elevated ketone levels, insulin's effects will take hold, causing inhibition of lipolysis and free fatty acid release and initiation of lipogenesis, protein synthesis, and glycogenesis. Now, this seems to act as a negative feedback kind of mechanism in people that have normal pancreatic function, as to disallow the production of overt quantities of ketone bodies, thereby preventing the ketoacidosis seen in diabetics. And since the provision of carbohydrates is minimal, there will be continual alternation of high and low glucagon and insulin levels to maintain the nutritional ketosis. So, what is the role of ketone bodies? And it makes sense that this occurs because ketone bodies have been found to be more metabolically efficient than glucose or fats in mitochondrial ATP production. So the functional tissues can be maintained because of the ketone bodies. So the brain, heart, and muscle can utilize these ketones. It's important to mention that although glucose is the favored energy molecule in the brain and other tissues, ketone bodies and fatty acids are the preferred substrates for neurolipid synthesis and cardiac function, respectively. Specifically, omega-3 fatty acids are preferred for neurolipid synthesis. After all, the brain is composed of docosahexaenoic acid, DHA, which is derived from omega-3 fatty acids. So foods with high omega-3 fatty acids are going to be nuts like walnuts, and also walnut oil, and also fish, tuna, salmon. Those are very high in omega-3s. And this whole fact that fatty acids and ketone bodies are preferred substrates for certain kinds of functions, this further supports the fact that ketogenesis is an ongoing normal biochemical process that is necessary for homeostasis. So if a ketogenic diet is implemented, will there be an added benefit, particularly in the brain? We shall see in a moment. But first, let's look how ketones are used by cells for energy. This figure shows a peripheral cell, for example, maybe a myocyte or a neuron, and it's using ketone bodies for energy production. Note that the beta-hydroxybutyrate is converted to acetoacetate once inside the mitochondrion, forming one NADH each time this conversion occurs. GTP is not able to be produced within the Krebs cycle due to the fact that the succinyl-CoA donates its coenzyme group to acetoacetate, creating succinate and acetoacetyl-CoA. Thiolase then cleaves the acetoacetyl-CoA to form two acetyl-CoA molecules. By the way, as a spoiler, exercise increases the amount of monocarboxylate transporters, which also helps transport lactate. Okay, so after all that we have seen thus far, one synoptic statement can be made. Ketone bodies are essentially vectors for acetyl-CoA. But how much energy is derived from ketone bodies? This table shows us the fact that ketones are energy sources and can act to provide a complementary energy source to fatty acids, as compared with one glucose molecule, which generally produces 20 ATP via aerobic means. Ketones are quite efficient energy molecules. However, there is something called ketoadaptation that must occur before ketones can be efficiently used as a primary energy source. So what are the four physiologic facts that are the basis for the evolution of ketoadaptation? Well, the body has a small reserve of glycogen, or preformed carbohydrate. The body has limited protein stores as well, because a lot of those proteins are in functional tissue. There is a relative wealth of stored triacylglycerols, as we can see in our obese and overweight population. And there is an inability of long-chain triglycerols, greater than 12 carbons, to cross the blood-brain barrier. So, how long does it take to adapt? That's a good question. The adaptation occurs within 3 to 4 weeks. And this time period essentially represents the full adaptation of nitrogen, fat, and carbohydrate metabolism. So brain adaptation most likely takes precedence here, especially in situations of energy adaptation. So let's have a look at the brain's metabolism. Well, the brain uses about 20% of all resting energy. And with normal carbohydrate provision, 100 to 120 grams per day of glucose are used, with the majority of ATP used for the maintenance of pre- and post-synaptic ion gradients and neuronal resting potentials. Hyperketonemia, which is high blood ketone concentration in the blood, must be substantial, greater than 0.2 millimoles per liter, before the brain can effectively use the ketones. The post-absorptive state, meaning after you consume food, and overnight fasts place ketone levels between 0.1 to 0.3 millimoles, which are much too low to jumpstart neuronal metabolism of the ketone bodies. And so astrocytes, which are glial cells within the brain, and they have ketogenic abilities. And so why do I mention the astrocytes? Well, because they are the most abundant cell in the human brain and have numerous functions within the central nervous system. And they include formation of the blood-brain barrier, glycogen provision and metabolic support, modulation of synaptic transmission, and regulation of ion concentration. And so they can use fatty acids and leucine for ketone body production. And so this is thought to be a survivalist quality, protecting the neurons from hypoxic-induced death. So this table shows how ketone body levels correlate with derived energy during periods of fasting and administration of a ketogenic diet. The remainder of the energy used by the brain is derived from the infamous obligate nutrient glucose. Recall that during lipolysis, glycerol is released from the 3 fatty acid tails. So this glycerol molecule can be used to produce glucose. The carbohydrates and glucogenic amino acids provided in the diet can render glucose as well, leading to glucose and glycogen not being abandoned. So you can't abandon glucose. You just can't do it. The information in this table is backed by PET scans of fasting mice or those fed a ketogenic diet, which showed a 7 to 8-fold enhancement of ketone body uptake within brain regions. Likewise, with advancing age and even in neurodegenerative disease, brain transport systems for ketone bodies have been shown to remain relatively intact. Thus, it seems that ketosis may be advantageous and may offer benefits to the brain. So how do ketone bodies benefit brain function? Well, in general, ketone bodies are considered to be neuroprotective, meaning they protect neuronal structures, and have been found to impart anticonvulsant properties. In rat models of brain hypoxia, anoxia, and ischemia, beta-hydroxybutyrate has been shown to improve neuronal function. Beta-hydroxybutyrate imposes protective properties against the Parkinsonism-inducing neurotoxin 1-methyl-4-phenyl-1,2,3,6 tetrahydropyridine, MPTP, in rat models. More specifically, MPTP causes degeneration of mesencephalic dopaminergic neurons, as seen in laboratory or experimental animals. And so beta hydroxybutyrate needs more attention. And the reason why this is so is because it restores substrates for mitochondrial electron transport chain. It decreases the reactive oxygen species via modulation of NADH and coenzyme Q levels in the mitochondria. The nicotinamide adenine dinucleotide, which is the NAD, is involved in redox reactions, and thus its modulation could promote favorable control of reactive oxygen species. It bypasses mitochondrial complex 1 via production of succinate, and this kind of goes back to the MPTP, because the MPTP actually causes degeneration of those dopaminergic neurons by inhibiting mitochondrial complex 1, which leads to apoptosis. The beta hydroxybutyrate overcomes this inhibition, like it states here, via production of succinate, thereby improving mitochondrial ATP production due to supposed improved oxidative phosphorylation. Beta hydroxybutyrate also increases electrical potential between mitochondria and cytoplasm. And it causes efficiency of the working heart to increase by at least 25% via augmentation and reduction of oxygen consumption. So this is kind of like a little prelude to what's to come for the possibility of the ketogenic diet with human performance and exercise. So, since we've discussed the ketogenic diet and the role of ketone bodies and the proposed benefits in the CNS, we now will turn our focus to an overview of studies that investigated the use of the ketogenic diet for neurologic disorders. Yes, the ketogenic diet has been investigated in clinical studies, epilepsy studies. Most children remain on a ketogenic diet for 2 to 3 years and are then weaned off the diet once their seizures have been controlled for about 1 full year. It is important to note that even though the children are on a ketogenic diet, they are still on anticonvulsant medication. So maybe the ketogenic diet is an additive effect or maybe a synergistic effect, but we can't say for certain that the ketogenic diet can cause reduction in seizures by itself. So the effect of the ketogenic diet on seizure prevention may be bolstered by anticonvulsant medication. That's essentially the consensus on it. And so we see that the therapeutic ketone concentration range is 2 to 7 millimoles per liter, which actually corresponds with that table that we saw. So at 7 millimoles per liter, there's at least going to be 60% of brain-derived energy from the ketone bodies, possibly up to 70%. There have been Alzheimer's disease studies as well, and the ketogenic diet has been shown to grant symptomatic relief, and the MCT administration has improved memory. So the MCT administration we talked about earlier too, being the modified KD, one of the primary variants. Parkinson's disease studies have also been studied, and a KD was shown to decrease the risk of development of the actual disease. And for Alzheimer's and Parkinson's disease, the increased amount of fat consumed in the KD may decrease disease risk because the amount of essential fatty acids that are generally within the diet. So we see here the KD has shown to improve resting tremor, freezing, balance, gait, mood, and energy levels in a small cohort of Parkinson patients. Of course, the KD has been used in animal studies. In epilepsy studies, spontaneous seizures have been attenuated in temporal lobe epileptic rats when the KD was used. Specifically, there was diminished hippocampal excitability and supragranular mossy fiber extension. So KD was shown to impart anticonvulsant and also anti-epileptogenic properties. In Alzheimer's studies, along with calorie restriction, a KD was shown to decrease amyloid beta deposition, as well as protect hippocampal neurons from toxicity due to amyloid beta deposition. Now, in Alzheimer's disease, a contradiction to clinical findings may exist here, but this may point to the fact that rats are not humans and vice versa, and that a certain type of fat incites this contradiction. Now, saturated fatty acids are implicated in Alzheimer's disease development, and high-fat diets are thought to increase amyloid beta peptides, which are the supposed culprit in the disease. So it might all come down to the kinds of fats and their percentage of the diet. In Parkinson's disease studies with animals, both calorie restriction and a KD have been shown to create resistance to MPTP-induced loss of dopaminergic neurons and protect against disease-induced motor deficits. In ALS studies, a KD was shown to be neuroprotective, and it slowed the progression of motor deficits. The ketogenic diet has also been shown to protect rats from neuron loss during insulin-induced hypoglycemia, and it can reduce up to 58% of cortical contusion volume at 7 days after cortical injury in rats. So what about starvation or calorie restriction? Recall that a KD mimics physiologic adjustments associated with starvation. Absolute starvation in rat models has been shown to diminish neuronal loss in brain regions like the striatum, neocortex, and hippocampus during a 30-minute 4-vessel occlusion. Starvation has also been shown to impart an anti-epileptogenic effect in rats. Thus, along with the ketogenic diet, calorie restriction could impart additive and maybe even synergistic effects so that there may be even greater overall effects. So overall, what does the KD do for the brain? It offers neuroprotection, resists neurodegeneration, it imparts anticonvulsant effects, and has anti-epileptogenic properties. But how does the KD impose such favorable effects? So there are several mechanisms responsible for the KD's neuroprotective effects, and that's what we're about to cover here. So, alterations in energy metabolism, glutamate-mediated toxicity, enhanced gamma-aminobutyric acid, GABA, production, alterations in apoptotic signaling, antioxidant effects, anti-inflammatory effects. First up are the alterations in energy metabolism. Carbohydrate restriction results in decreased availability of glucose and subsequently lowered insulin, which leads to the induction of stress proteins. This induction lends to diminished reactive oxygen species, stabilization of intracellular calcium, and maintenance of mitochondrial function. Beta-hydroxybutyrate may provide a more efficient source of energy per unit oxygen for the brain as compared to glucose. The KD upregulates hippocampal genes for energy metabolism and mitochondrial enzymes, and mitochondrial biogenesis and ketosis lead to neuronal resistance to metabolic stress. So, the number of mitochondria increase due to the adaptation to the ketogenic diet. The glutamate-mediated toxicity. Well, acetoacetate is thought to protect against toxicity induced by excessive glutamate. Excessive glutamate results in the induction of ROS, so technically acetoacetate may inhibit that induction. Enhanced GABA production. Well, the KD increases GABA concentrations in rats and humans. GABA-mediated inhibition is thought to be neuroprotective. Then GABA has functions for synaptogenesis, neurotransmission, and neuronal protection. Alterations in apoptotic signaling. The KD is protective against apoptotic cell death caused by glutamate receptor agonists and excitotoxins. Specifically, the KD increases calbindin, which is a protein responsible for buffering intracellular calcium, which leads to decreased apoptosis, thereby granting neuroprotection. Also, the KD decreases cholesterol, which is a pro-death signal that is activated by excitotoxins. Antioxidant effects, everyone's favorite effects. Ketone bodies have been shown to decrease the amount of coenzyme Q semiquinone, which prevents the production of ROS. Now, some controversy surrounds semiquinone as to whether or not it functions as an oxidant or antioxidant. However, portions of the semiquinone molecule do exhibit a likelihood for free radical production. Ultimately, the KD likely accomplishes the increased antioxidant effect via semiquinone diminishment, whereby fully reduced and/or oxidized forms of the enzyme are formed for their participation in antioxidant systems and electron transport chain. The KD increases the concentration of glutathione peroxidase in the hippocampus of rabbits. Also, the KD increases uncoupling proteins in mitochondria, leading to decreased ROS production, especially within the dentate gyrus. Anti-inflammatory effects, also popular effects. Inflammation has been hypothesized to contribute to the development of chronic epilepsy. The fatty acid loads provided by the diet will lead to activation of peroxisome proliferator activated receptor alpha, or PPAR alpha. Inhibition of transcription factors will result from this activation, which will cause an inhibition of nuclear factor KD and activation protein 1. When these transcription factors are unregulated or unsuppressed, it essentially will result in an inflammatory cascade that have been associated with inflammatory and autoimmune diseases. Now, up to this point, we've only discussed the positives or benefits associated with a ketogenic diet. Although very few serious negative effects exist with this diet, they nevertheless should be mentioned. So what are the negative effects associated with a ketogenic diet? Well, high fats increase the risk of cardiovascular disease and events, and even certain cancers. You know, high fat diets have been the subject of much controversy and are still inconclusively linked with cardiovascular disease and cancer. Conversely, a high carbohydrate, low fat diet has been shown to increase fasting triacylglycerols and decrease HDLs, the good cholesterol, which is the perfect concoction for enhanced atherogenesis or plaque formation. High fat diets may increase the risk of AD development, Alzheimer's disease. Now, the development of Alzheimer's may be based on high saturated fats, but most ketogenic diets typically are made up of 80% poly and monounsaturated fats. So, depending on the fat intake, that might actually increase or decrease the risk of Alzheimer's disease development. Cholesterol increase by 30% is possible. Now, genetic differences in handling of dietary lipids may predispose some individuals to an increased total blood cholesterol. However, the HMG-CoA reductase complex responsible for ketogenesis is also associated with cholesterol synthesis. Loss of lean body mass and cardiovascular reserve capacity is possible, but if electrolyte balance is normalized and protein intake is adequate, this should not occur. We'll get into a little bit more on this later in the human performance portion of the presentation. Hepatic stress may be involved because if the liver can't truly utilize ketones for energy, then this may place the liver under some sort of metabolic stress. Skeletal wasting is possible. Bone demineralization is possible with acidosis, but the low-grade acidosis induced by a ketogenic diet pales in comparison to the acidosis seen with diabetic ketoacidosis. Nephrolithiasis or ketone kidney stones are usually a complication seen in the pediatric population. but treatment with potassium citrate can inhibit much of the stone formation seen with the ketogenic diet and anticonvulsant pharmacologic therapies. Micronutrient deficiencies, which we discussed very early on, these deficiencies are likely if insufficient amounts of fruits and vegetables are not consumed. Now, if we recall the ketogenic diet food pyramid, there were mainly non-starchy vegetables and fibrous fruits. So many B vitamins are found in breads and cereals, which are banned foods in a ketogenic diet. We will see more on nutritional strategies that fit into the ketogenic diet later in the presentation. Dehydration may occur, mainly due to the fact that decreased water intake by up to 80% used to be recommended, which is no longer the case. Also, since there is a relatively large intake of protein, this could lead to diuresis and possible dehydration. Behavioral changes are possible, and they may occur due to the metabolic changes imposed upon the reward system in the brain, as well as the changes in energy type may alter behavior during the keto adaptation phase. Vomiting is possible, and amenorrhea, the cessation of menstruation, are possible but not common with the ketogenic diet, but they have been documented as possible side effects. This is a summary of pros and cons related to a ketogenic diet, and so I'll just let you all read over these, but as you're reading, I'm going to say one thing, and that's this concludes the first portion of this presentation. The next portion will consist of oxygen toxicity, relating them to seizures, and then going over how the ketogenic diet may decrease the risk for oxygen toxicity induced seizures. So stay tuned.